Hi, my name is Aseel Rawashdeh, and I'm a student at Anderson High School in Austin, Texas. And in my project, I synthesized a new mosquito larvicide by loading common household essential oils into Saccharomyces cerevisiae or Baker's yeast cells using no synthetic chemicals or inaccessible equipment. I did this because mosquitoes are the most dangerous animal on Earth. They kill over 1 million people every year and cause hundreds of millions of cases of long-term illnesses like paralysis, microcephaly, and developmental delays. The problem here is that these illnesses currently don't have many treatments. And as a result, we rely almost entirely on mosquito vector control or population management in order to prevent these diseases. Synthetic larvicides, most commonly used and studied, are very toxic to ecosystems and even humans, and they rapidly select for resistance because they all target very similar action sites. Now, you might be wondering, why did I use yeast to make my larvicide? Well, yeast encapsulation was a relatively popular method of carrying dyes, flavors, and pharmaceuticals in the early 2000s. Additionally, this study was inspired by a similar study on termite treatment encapsulated in yeast to prevent termites. Now, in this case, yeast is advantageous because it stabilizes the volatile essential oil in a protective shell, and it has a high loading capacity, and mosquito larvae prefer to consume the yeast, creating a Trojan horse effect. Additionally, most aquatic insects and invertebrates cannot digest the complex beta-glucans in the yeast cell wall, which prevents non-target toxicity. In my experimental larvicide assessments, the three essential oils that worked were cinnamon, garlic, and orange oil. And I completed about 700 total bioassays throughout my research. There were four main findings. First, I found that the yeast microcapsules exhibited high larvicidal activity at very low concentrations. The LD50, or the lethal dose 50%, was between 20 to 35 parts per million for all three of the larvicides. Additionally, all of the larvicides also inhibited pupation in surviving treated larvae. Notably, for cinnamon treated larvae or cinnamon loaded yeast treated larvae, 100% of surviving treated larvae did not pupate in concentrations as low as 20 parts per million. Second, many morphological and behavioral alterations were observed, including melanization, neural excitation, neural inhibition, midgut and tracheal damage, and expulsion of Malpighian tubules and cuticle blebbing. Five of these did not occur in the essential oils alone, and three were previously unreported in the literature, suggesting the discovery of new target sites which could help guide future research in this area. This exploitation of multiple target sites is significant because it prevents the rapid selection of resistance, which is very advantageous for the long-term use of this larvicide. Third, I found that all three treatments had no lethal effects on water fleas and green microalgae. These results suggest that the new larvicide will have low non-target toxicity and will not affect ecosystems negatively, although more testing must be done in integrated ecosystem models to confirm this. Fourth and finally, I tested my larvicides on Toxorhynchides retilis, which is a beneficial non-hematophagous mosquito, which basically the larvae of this mosquito eat the larvae of dangerous mosquito larvae, making them a potential biocontrol agent. Now, I found that all three of my yeast-based larvicides had no toxic effects on Toxorhynchides because these larvae are carnivorous. However, BTI, the positive control, killed all of these larvae. Now, this is really exciting because this is the first available larvicide to have no toxic effects on this beneficial species of mosquito, making it a potentially very valuable and integrated mosquito management. To conclude, this novel yeast-based larvicide helps lower the human and economic costs of mosquito-borne diseases in endemic regions in countries like Brazil, the Philippines, India, and South Africa, where mosquito treatment methods can be costly or ineffective. This larvicide presents multiple advantages, including high efficiency and efficacy, low cost, low non-target toxicity, biodegradability, and the prevention of resistance evolution. By integrating this larvicide with existing mosquito control methods such as genetic modification and biological control, humanity can be one step closer to eliminating the deadliest animal on Earth.